Okay, we might begin now. So I'd like to welcome everyone to this ACUR at ANU presentation session, Cancer Research Visualised. Before we begin, we'd like to acknowledge and celebrate the first Australians on whose traditional lands we meet and pay respects to the elders past and present. My name is Aidan Delaney and I will be the chair for today's session. Our first presenter for today is Yongyan. So please start when you're ready.
Hi everyone, I'm Yong and today I'm going to present to you my honours project, which is about understanding dendritic cell immune checkpoint proteins in cancer immunotherapy. So I'll just provide some of the backgrounds. So PD-1, which stands for programmed cell death protein 1, is an important immune checkpoint protein in our immune system. It's normally expressed as a cell surface receptor on T cells, and its function is to inhibit T cell activation during an immune response. So an example shown in this schematic here, so that our immune system can be balanced and avoid hyperactivity. And one of the most important source of PD-L1, which is the ligand that binds to PD-1, it's from dendritic cells. And dendritic cells can present foreign antigens to T cells and activate T cells so that the T cells can fight foreign cells or tumor cells based on the antigen they got presented. However, a lot of tumor cells also mimics this system. They express ligands for PD-1 as well, and they suppress T cell activation shown in this schematic here. So targeting this PD-1 pathway is an important approach for cancer treatment. And in fact, there has been a lot of PD-1 inhibitors developed for cancer immunotherapy, but it is still failed in a lot of cancer types, and the exact reason remains unknown. So for my honours project, I'm trying to address two questions. So first, can blocking this T cell checkpoint molecule improves nanoparticle vaccination? And secondly, what are the molecular pathways that regulates PD-1, PD-L1 binding? So I'll first introduce my first question, which is about nanoparticle vaccination. So polyhydroxyalkanoates, which stands for shorthanded PHA, is a naturally produced polyester by a lot of bacteria, and they produced it as carbon source storage under starvation conditions. And it's biodegradable, biocompatible, and it has ability to enhance the immune response. So in this project, we conjugated ovalbumin peptide with PHA beads to generate what we call O3 beads. So ovalbumin is a chicken egg white protein that is recognized as foreign by the mice immune systems. And dendritic cells can then present this ovalbumin to T cells and activate them. So the point is to generate a strong enough immune response so that foreign cells or tumor cells can be killed by those activated T cells. So in this experiment, we want to see if the O3 beads is able to generate an immune response in mice. So first, mice are immunized by O3 beads to generate an immune response. We then inject cells that are labeled and can be traced to see how many cells got killed. And this gives us an idea of how strong the immune response is. And as we can see in this graph, there is a immune response generated by the O3 beads. So next, we combined with anti-PD-1, which is PD-1 inhibitor with O3 beads, to see if the PD-1 inhibitor can improve this nanoparticle vaccination. And we predict as we block this signal, as we block the PD-1, more T cells will be activated, so there will be a stronger immune response. So as we see in this graph, we do see a stronger immune response in combination with PD-1 inhibitor. And then we want to know if this has an effect in T cell proliferation, since more T cells will be activated, so maybe T cells will proliferate more. So in this experiment, we also have two groups, but because of the time being, I would briefly just describe the result without talking about the detail of the methodology. So we have two groups here. So just the O3 beads alone, and also O3 beads with PD-1 inhibitor. And we do see T cell proliferation in those two groups, but there's no significant differences between those two groups. So this might be because the O3 beads itself is able to generate a sufficient immune response for the T cells to proliferate. Next, I will talk about my second question, which is about the molecular pathway that regulates PD-1, PD-L1. From a CRISPR-Cas9 screening experiment done previously by our lab member, we defined some candidate genes that might potentially involve in PD-L1 regulation. So this involves BCL6 and BCOR genes. BCL6 gene encodes a transcriptional repressor that is important for germinal center formation, and BCOR can interact with BCL6 to assist BCL6's function. And they are candidate genes that may involve in PD-L1 downregulation. So maybe these two genes are part of a transcriptional regulator that can interact with PD-L1 transcription. And to confirm this, we perform single gene CRISPR-Cas9 knockout in murine dendritic cells using DC1940 cell lines. So deletion of those genes should increase PD-L1 expression. And again, in the meantime, I won't go through the detail of the methods here, but we've successfully generated cells with BCL6 deletion and BCOR deletions. So with the BCL6 knockout cells and BCOR knockout cells, we do see an increase of PD-L1 surface protein level as we expected, shown in this group 2 quantitative graph here. And you can also see this wave graph here. And BCL6 knockouts also results in a total increase of protein levels in both resting state and interferon gamma stimulation states, shown in this Western blot. So just note that we activated dendritic cells using 2 different stimulants to see the effect of the knockouts. So first is interferon gamma, and which is a self-secreted cytokine that is known to have a role in anti-tumor immunity. And second, we use CpG activation, which is a molecule that mimics bacteria stimulant. So the cells will be able to activate it, and the activated state is where it recognizes foreign antigen and then present to T cells as we described before. So as we see in those 2 graphs, upon both interferon gamma and CpG activation, we see an increase of PD-L1 surface expression as we expected. And interestingly, if we look up this graph here, we also see FLT3 expression is significantly reduced in BCL6 knockout cells, and also in protein level as shown in this plot here. So FLT3 is a very important receptor that is for development of disease. So maybe BCL6 and FLT3 are somehow indirectly linked and regulate in conventional dendritic cells. And in conclusion, BCL6 and BCORs are evidently involved in PD-L1 regulation in dendritic cells, and we propose a mechanism in here. And moreover, BCL6 may involve in FLT3 expression in dendritic cells. And some of the future direction, we can reconstitute the wild type genes back into the knockouts to validate our results. And also for our nanoparticle question, we can perform tumor assays to see the combination treatment of O3 beads and anti-PD-1 can improve tumor clearance. Thank you very much for listening.
Fantastic. Thank you very much for that presentation. So we now have 5 minutes for Q&A. I'll begin by inviting the judges if you have any questions.
Melissa, any questions from you, or would you like me to start us off? Okay. Firstly, I just want to say thank you so much for a wonderful presentation. I will be fully transparent that I have very little understanding of biology, let alone cancer research, and the fact that you were able to communicate your research in a way that even I was able to understand, I think was a really valuable skill and an excellent kind of comment on the quality of your work. So congratulations on that. I do have a couple of questions that I am just curious and perhaps a bit more interested in, but could you firstly just contextualize, I guess, the tradition of research where this kind of sits at the moment? You mentioned that you had that somebody else in your lab had done previous work on this. Is this early experimental? Is it a little bit further down the line?
Actually, this immune checkpoint protein is kind of at the state of clinical application at the moment, but there is still a lot of issue about it. In fact, I think it was 2018, there was a Nobel Prize even awarded for PD-L1 regulation research because they actually transform this kind of research into actual clinical practices, and a lot of patients actually benefit from it. But in the meantime, when they still like doing this, and a lot of patients are still either not responsive or the results aren't really good. So doing this kind of research at the moment, so we're trying to resolve these issues. Yeah.
Fantastic. And that actually leads quite nicely into my second question, which is, again, I'm just wondering if you could...
Sorry, you cut out.
Is it working now?
Yeah.
Yeah? Oh, okay. Sorry about that. But yeah, sorry. So my second question is, I'm wondering if you could comment on some of the future work that I guess is lined up in order to continue validating this line of research.
Yeah, actually there's a lot of experiment can be done on these immune checkpoint proteins. So, in here I mentioned our lab member has previously do a genome-wide screening. So a genome-wide screening is, so you basically, you want to screen your the whole genome of a mouse cells, for example, and you're trying to find which gene is actually related or regulate this particular protein, so PD-1. So you don't know what you come out as a result. So he actually generated a lot of data, and he found quite a few genes that may be involved in this protein regulation. So I'm just working with one of them, but there might be a whole lot of genes that is regulating this protein. So there might be a massive gene regulation map. And actually working out this mechanism is very important, because in some patients, so everyone's genetic is different, and working out maybe some patients main problem is a particular gene and the other patient is a particular gene, but you work out this whole map so you have a more clear understanding of the PD-L1's regulation. So I guess it will have a massive improvement in understanding why some of the patients won't respond to the treatment.
Perfect. Thank you so much for that. Those are all of my questions.
Thank you.
Excellent. Were there any more questions from the judges? If not, we have a brief amount of time. If there are any audience questions, you can raise your hand and we can unmute you, or pop them in the chat. Give it a few seconds for anyone if you have questions to come forward. Okay, it doesn't look like we have any further questions, so we might finish that presentation. Thank you very much for that. And we'll move on to our final presentation for today's session. So our final presenter is Nohad Maroun. If you'd like to commence when you're ready.
Yeah, hi. Sorry, my dog's just crying in the back, so just ignore that. Okay, does everyone see my screen? Yes, great. Okay, so my name is Nohad Maroun. I'm an honours student at the University of Sydney. I'm completing my honours at the Children's Medical Research Institute at CMRI in Westmead. And my study this year looked at actin and its role in telomerase recruitment to tumors. So cancer is a neoplastic disease, and it develops from the ongoing accumulation of mutations that lead to the disordered proliferation of somatic cells. Replicative immortality is a crucial hallmark that cancer cells possess as normal cells undergo cellular senescence. And that's a process that limits the proliferative ability of cells reaching their replicative limit. And this is mediated by telomere shortening. So what is a telomere? Telomeres are the protective caps that are found at the end of chromosomes. They're repetitive TTA GGG repeats, and they shorten every time the cell undergoes division. So once the telomeres of cells reach a critical length, normal cells undergo cell senescence, or in other words, the cell stops dividing. However, cell senescence can be counteracted by telomerase, which is an enzyme, and it lengthens telomeres by adding the telomeric sequence repeats. Thus, the cell can continue to divide, and this leads to the uncontrolled proliferation of cells and tumorigenesis, which is the formation of tumors. Now, telomerase, the enzyme, is present in approximately 85 to 90% of all cancers. Thus, it's a potential therapeutic target for cancer. But there is a gap in the knowledge regarding how telomerase gets to the telomeres, or how telomerase is recruited to telomeres. So recently, our lab has identified a link between telomerase recruitment and the DNA damage response. So the DNA damage response has two kinases, and a kinase is a molecule that phosphorylates other things. These two DNA damage response kinases, known as ATM and ATR, are the master regulators of the DNA damage response. Now, ATR is known to mitigate replication stress, which is any process that hinders or pauses DNA replication. And our lab has actually found a link between the ATR-mediated response to replication stress and telomerase recruitment to telomeres. So the major finding was that initiating replication stress increased telomerase recruitment to telomeres. Concurrently, there were studies taking place at the Caesar lab, which is also at CMRI, that showed the formation of nuclear actin fibers in response to replication stress. And actin is an abundant protein that's involved in many cellular processes, such as muscle contractions, cell division. It also forms the cytoskeleton of the cell. So actin monomers polymerize, and they form filamentous actin or F-actin. And our lab has generated preliminary data that has linked F-actin to telomerase recruitment. So that brings me to the aim of my study this year, which was to investigate the involvement of actin and actin-related proteins in telomerase recruitment to telomeres. So our hypothesis is based on a pathway. So we hypothesized that replication stress activates the ATR kinase, which leads to actin polymerization and telomerase recruitment to the telomere. Now, there are two other actin-related proteins that I investigated in this study. Lim kinase, which is a negative regulator of actin polymerization. So it phosphorylates cofilin, which is a protein that cleaves actin filaments. So that frees actin monomers. In other words, it breaks the actin filaments apart. So Lim kinase inhibits this process. And myosin is a motor protein that transports various cargo along the actin fibers. So in order to investigate my aims, we used fluorescence in situ hybridization, or FISH, which is a modified fluorescence microscopy technique, and it allows us to detect and locate specific DNA sequences on a chromosome. So we probe for telomeres, which is shown in red here, and we also probe for the RNA subunit of telomerase, which is called HTR, and it's shown in green. We use a DAPI nuclear stain in blue, which identifies and allows us to count nuclei. And we also use EDU, which is a nucleotide analog for uridine, and this is incorporated into the DNA of replicating cells. And that's shown in pink here. So this allows us to identify S-phase cells, and this is important because telomerase is more active in S-phase cells than any other phases of the cell cycle. So after we imaged these, we quantitated the co-localizations between telomeres and HTR. So the co-localizations is the events where the telomeres and the HTR overlap. So that indicates the presence of telomerase over telomere. So we combined this fish with chemical inhibitors against actin polymerization, limb kinase, and myosin. And we also used a phytocolin to cause replication stress. And just a side note, unfortunately, due to the Delta outbreak in Sydney, I was only able to complete 2 repeats for each of these experiments. However, we first looked at actin polymerization as a whole, and we wondered whether inhibition affects telomerase recruitment. So we treated 293 T cells, which are an embryonic kidney cell line that are highly transfectable and are also very high in telomerase foci, with latrunculin B. And latrunculin B is an inhibitor of actin polymerization. So it works by binding to actin monomers, which prevents them from polymerizing and forming the F-actin. And we also use DMSO, which is dimethyl sulfoxide, as the control. It just freezes the cells and prevents them from drying during the experiment. The y-axis here denotes the number of HTR and telomere foci that have overlapped or co-localized, as I said, in the S-phase cells, so the EDU positive cells. So looking at the panel, latrunculin B treatment resulted in a significant decrease in telomerase recruitment compared to the control DMSO sample. This shows us that actin is involved in the telomerase recruitment pathway. So moving on to the second panel, it shows a series of treatments that were performed on 293 T cells. We inhibited limb kinase, which I described before as a regulator of actin polymerization, using limb KI. We also inhibited myosins using the 2 inhibitors, BDM and BTS. We also initiated replication stress using a phytocolin, which is a DNA polymerase inhibitor. Then we performed individual inhibitor treatments and dual treatments with the inhibitors and a phytocolin. So as we can see in the figure, the significant increase in co-localization seen in the phytocolin treatment is consistent with previous findings that replication stress increases telomerase recruitment to telomeres. So compared to the DMSO control, the individual and the dual inhibitor treatments all showed a significant reduction in co-localizations. Moreover, the increase in telomerase recruitment that was seen as a result of the phytocolin-induced replication stress was abrogated by the inhibition of limb kinase and myosins. So we can conclude from these results that the actin-related proteins, limb kinase and myosins do play a role in telomerase recruitment to telomeres. So overall, the inhibition of actin polymerization and the actin-related proteins resulted in reduced telomerase presence at telomeres, which suggests that actin and its regulators are important for telomerase recruitment. There are many future directions for this study, such as looking at other actin-related proteins, including cofilin, which severs the actin filaments, which I described before, important, which imports actin into the nucleus, and formin, which is responsible for polymerizing actin filaments. We could potentially inhibit or knock down these proteins and observe their effect on telomerase recruitment. Also, we will be performing these studies on other cell lines, such as HeLa, which is an immortal cell line that's very commonly used, in HCT116 cells, which are a colon cancer cell line that's also telomerase positive. Currently, we're looking at doing live cell imaging because these experiments are done on fixed cells. So live cell imaging will allow us to actually visualize telomeres and telomerase and their interactions with actin in real time. So if actin is associated with telomerase recruitment, we could potentially see the movement of telomeres and telomerase along the fibers. Also, we know that telomerase is present in stem cells and cancer cells. So our lab is currently doing research on telomerase action in stem cells, as stem cell telomerase may function under different pathways compared to differentiated cells. Thus, identifying the telomerase recruitment pathway present in cancer cells will possibly inform therapeutic design of telomerase inhibitors. So ultimately, this study will aid in unraveling unknown components along the recruitment pathway and will contribute to the understanding and possible targeting of telomerase function in cancer cells. Thank you.
Fantastic. Thank you for that presentation. Okay, again, we'll now have 5 minutes for Q&A, and so I'll start by inviting the judges if you have any questions.
I jump in first this time, Natalia. Thank you so much, Nohad, for your presentation. I really enjoyed that and looking through your poster as well. It's really clearly presented, and I love your figures there. As you were speaking, I was wondering about the impact of COVID on the research in your lab, and you answered that. You mentioned being able to do only 2 experiments. What would be the kind of typical or the preferred number of experiments you'd want to do in this kind of research?
Okay, well, with these experiments, you are expected to do at least 3 repeats because then you can do statistical analysis on the experiments. However, with being a scientist, you need to do a lot more than 3 repeats. So I've actually completed the experiment quite a number of times just to try and optimize it. But the best looking experiments are the ones that I had to include. Of course, yeah, there was a period where I was performing about 3 weeks in a row the same experiment, but I had to optimize the actual experiment. Yeah, so maybe around, well, 3 is the cutoff. You have to do minimum of 3 experiments. Yeah.
Great, thank you.
That's all right.
Fantastic. Again, I definitely echo the same sentiment. It was a wonderful presentation and a great poster to kind of follow through with. I think my question, admittedly, is perhaps broader than what you're usually faced with, but I think it is very related to the fact that recently, in the last 6 months, I've also been doing some interesting, that I find interesting, research out at the Children's Hospital of Westmead as well. And I've been working with the nurses and have been very close with the people who have been entering and leaving the hospital. Yeah. And broadly, I think I would just offer thanks and also congratulations to everyone who's been doing research in a medical space. It's always wonderful to have people who are, you know, striving to improve quality of life and health for everybody else. And I think my question just kind of comes down to this idea of translating your research from a context like this, or a context with your supervisor and your actual lab team, to the potential patients and carers who are in the hospital itself. So I'm wondering, if you were to communicate this research to somebody who was a patient or a carer, what kind of reassurance would you be able to offer them?
Okay, well, that is a tough question. The reassurance that I'll be able to offer them, because we do know that cancer is it's kind of like a personalized disease. So it's different in every single case you get, right? So because telomerase is present in a high number of these cancers, so 90% of cancers, this is very promising. So if we can actually find a way to target telomerase directly or indirectly, hopefully through these studies, this could be very, very beneficial for a lot of people. So we could actually use, I would tell people, I'll tell patients, we could be using telomerase inhibitors in conjunction with already existing chemotherapies or other targeted therapies as well. So that would be the outcome of this study, but I would definitely offer that kind of reassurance to patients.
Perfect. Thank you so much. I know that was definitely a bit of a tough question, but that's a great reassurance to have. And again, all the best to you and your lab and your research team for continuing this work.
Thanks so much.
Thank you. Okay, so we do still have a minute remaining if there are any audience questions as well. I think we have a raised hand here. So I'll just go ahead. Yongyan, if you'd like to. Yeah.
Yes. Oh. Sorry. I actually learned quite a lot of genetics about telomerase when I was in like it's my major.
Oh, really?
Yeah. I'm just curious because I know like in normal cells there's also telomerase. That's like the reason why we're getting old and like not functional with the normal cells. But like if you want to target telomerase in cancer, how would you kind of separate, you know, like the healthy part of cells telomerase and apart from the cancer ones?
Okay, so that's a great question. Like you said, there is telomerase present in normal cells. However, the number of telomerase is very, very, very low in normal cells. So it's basically, you know, it's close to zero. Because in cancer cells, it's present at around 100-fold foci.
Yeah.
Yeah. So if we actually find, the reason why I'm looking at the way telomerase is recruited is I'm trying to find a way to target cancer cells specifically and also in replication stress. So this is a thing that happens commonly in cancer cells. So this process happens in cancer cells. So that's why we're looking at replication stress and actin, because it's specialized to cancer cells.
Okay.
Yeah. So now what we need to actually find is we need to distinguish between cancer cells and stem cells, which is a study in the lab as well.
Oh, okay.
Yeah, so that's...
Right. Sorry to interrupt, but we are at time there. So for fairness, we might finish up there. But thank you very much to everyone for those presentations. So that does bring us to the end of today's session. I'd like to thank our judges, Associate Professor Melissa Davis and Natalia Gulbranson-Diaz, and also thank our student presenters. Congratulations on your presentations. So this recording will be made available online after the conference, and we hope to see you at other conference sessions this week. Thank you very much, everyone.